these are all replaced. Taste is a sense that we use every single day to tell us the things that we do like, to tell us things that we don't like the flavours of, to tell us what's good for us, and to tell us what could harm us. It would be pretty dull therefore if we couldn't taste. So how does this actually happen?
The tongue is the main organ of taste. On its surface are many small bumps called papilla, which are smaller at the tip of the tongue and then larger at the back. Note that there are 4 different types of papilla: phalate, filiform, foliate and fungiform. Not visible to the naked eye are small taste pores between papilla. Here is a close-up image of a papilla, shown here, and between you can see the taste pores. Along the edge of each taste pore are these taste buds. Each papilla can contain between 1 and several hundred taste buds. This is a close-up image of a taste bud. Within each taste bud there are between 50 and 150 taste receptor cells, grouped together like segments of an orange. These taste cells connect to gustatory afferent axons to transmit a taste to the brain, so we become consciously aware of the flavour. Basal cells are also present in the taste bud to produce new taste cells as older ones wear away. This diagram shows the basic mechanism taken by all tastings behind sensation of taste. Firstly, a cation enters through a channel in the taste cell. This leads to taste cell membrane depolarization in response to the tastant. If this depolarization is large enough, an action potential will be transmitted through the cell. This will cause voltage-gated calcium ion channels to open, calcium influx into the cell, and then release of a neurotransmitter from synaptic vesicles to the gustatory afferent axon, which will then fire action potentials signaling taste to the brain. The way in which taste cell depolarization occurs, though, differs according to which one of sweet, sour, salt, bitter, and umami flavours is being sensed. This is the way in which a taste cell detects salt. The main chemical in food giving it a salty flavour is sodium ions, and these are detected by the taste cell. As we eat salty foods, sodium ion concentration increases outside of the cell, then diffuses into the cell through sodium ion channels down its concentration gradient. The taste cell will then depolarize, and if this is large enough, fire an action potential to be transmitted to the gustatory afferent axon. Sour tastings are sensed in a similar pathway. Sour foods have a high hydrogen ion concentration, meaning a low pH. This is what gives a sour flavour. Hydrogen ions will dissolve in water and increase in concentration outside of the taste cell, as sodium does when salt is being detected. However, there are 2 ways in which protons can lead to depolarization. They can either permeate the sodium ion channel to enter the cell, or they can block potassium ion channels, so potassium becomes trapped inside of the cell, increasing the intracellular positive charge. Both of these routes will then lead to depolarization. The 3 tastes of bitterness, sweetness, and umami are transmitted via the same intracellular pathway. They all bind to G protein-coupled receptors to activate the taste cell. The tastings will bind to the GPCR to activate the intracellular G protein, which then activates phospholipase C. Phospholipase C will break down phosphatidylinositol 4,5-bisphosphate to diacylglycerol and inositol triphosphate. Inositol triphosphate is the important second messenger we are concerned with in taste. It will bind to a taste cell-specific ion channel, allowing sodium ions to enter the cell. This will lead to depolarization and calcium entry through voltage-gated calcium channels. Inositol triphosphate can also trigger calcium release from storage sites within the cell. Both routes will lead to neurotransmitter release, and hence gustatory afferent axon depolarization. So how are these 3 flavours not confused for each other if they take the same intracellular pathway? Bitter molecules bind to GPCRs encoded by the T2R gene. Sweet molecules bind to GPCRs consisting of 2 proteins rather than 1, as in bitter GPCRs. These are encoded for by T1R2 and T1R3. If one of these is mutated, an animal may not be able to perceive a sweet flavour. Umami GPCRs are also composed of 2 proteins. These are encoded for by T1R1 and T1R3. These taste receptor cells containing these proteins also connect to different sets of gustatory afferent axons, meaning these flavours can be transmitted individually to the brain. Let's just recap the pathway tastants take from the tongue to the brain. First, they are detected by taste buds in the tongue. Primary gustatory axons then carry the signal to the gustatory nucleus in the medulla. From here, the signal is transmitted to the ventral posterior medial nucleus within the thalamus. Then, the signal is transmitted to the cortex, so we become consciously aware of the flavour. There are 3 main cranial nerves involved in transmitting the signal of flavour from the tongue. These are the facial nerve, cranial nerve 7, transmitting the signal of taste from the anterior 2/3 of the tongue, the glossopharyngeal nerve, cranial nerve 9, transmitting the sense of flavour from the posterior third of the tongue, and then the vagus nerve, cranial nerve 10, transmitting taste from the glottis, epiglottis, and the pharynx. All fire to the brainstem, where they bundle together and synapse in the gustatory nucleus of the medulla. This is part of the solitary nucleus. From the solitary nucleus, gustatory axons then fire to the ventral posterior medial nucleus of the thalamus. Neurons here coordinate sensory information from the head. The next step in the pathway is the firing of neurons to the primary gustatory cortex, located in Brodmann's area 36 and the insular cortex. When a sense is transmitted to the cortex, we become consciously aware of it. The tongue is not the only transducer of flavour. Combinations of taste and smell contribute up to 3/4 of what we think is taste alone. To demonstrate this, we are going to perform a simple taste test, asking our volunteer to distinguish between 2 different foods when she is blindfolded and has her nose clipped. This is our volunteer. Hi! Now our assistant is blindfolded. Here is sample 1 and here is sample 2. Julia? Yeah, open. What do you think that it tastes like?
It could be apple or maybe a pepper or something.
And now to try sample 2. Okay, Julia?
Yeah.
What do you think it tastes like?
I'm not sure, it could be apple again or something.
The taste test just performed demonstrated that the tongue is not the only organ involved in taste, and it is in fact a much more complex process. In addition to olfaction, touch, in particular temperature, is also known to play a role in taste. Furthermore, a recent study by the University of Copenhagen found that a decrease in the ability to taste was correlated with congenitally blind patients. The taste receptors covered so far each respond to a particular flavour. It is important to mention that in reality there are many more flavours than the 5 covered, each a combination of the ones we've mentioned. So how are these in-between flavours detected? Receptors can be described as broadly tuned, meaning they can respond to more than one flavour. Furthermore, taste cells responding to different flavours can also converge their input to signal the wide range of flavours. We hope you've enjoyed our video on the mechanism of gustation.